Mommy, eh? Okay, okay. All right. Good morning, everyone. Again, can you confirm that you can hear me? If you can hear me, type. What should you type this morning? Cell, C-E-L-L, not clearly. Some people are selling. Oh, okay, great, great. So we have about 5 minutes more. I'm just going to share my screen and allow more people join so that by 8, exactly we would start. We still have space on this Zoom, so I'm hoping that people are not on the YouTube channel, hoping that the Zoom room is filled up, but it's fine. If the attendance is very low today, I would use the attendance, because sometimes I know that some of you don't join because we're not using attendance. So please put it in your class group. If the attendance is low, I'll be on the lookout. And I could just classify by department and score. So I could say maybe physiology, 2 marks, pharmacy, 3 marks, depending on the absent to present ratio. Okay. So let me just share my screen. All right, can you see my screen? If you can see my screen, type. No, don't write your name in the chat box. Don't worry. I will pull it from Zoom report. But if you can see my slides, type nucleus. Nucleus, if you can see my slides. In the chat box. Okay, good. So can somebody help me also ask those on YouTube, because now I've projected, or I can actually use my phone to see. Okay. Okay, I think they should be able to see it because I can see from my phone as well. All right. I'm just going to start recording. Please stop your video. Again, I just couldn't figure out how to turn off video for everybody. Maybe there's a setting somewhere. Video settings. Let's see. It's only my own. Allow. Okay, I found it. Yep. Oh no, it's not this one. Okay, I mean, I couldn't figure it out, but let's just move on. So the time is 8 o'clock. I would just start recording. Right. Okay, so in our last lecture, we started looking at the topic cell structure and function, and I think we stopped here. All right, the Zoom room is full. We have 300 people now. Great. And I said that one could think of the plasma membrane as a gate, that controls what goes into and out of the cell. I put up this picture, but we didn't have enough time to explain it. We're just going to look at the functions of the plasma membrane as specified on this image, or in this image. So we have the first one being the boundary and permeability barrier. And from secondary school, when a membrane, when they say, oh, through a semi-permeable membrane, What that means, permeability has to do with ability to allow substances go through selectively. So it selects what goes in, selects what goes out. And that's why the membrane is referred to as a permeability barrier. So it serves as a boundary. So when you have the membrane, some organelles have more than one membranes. The outer membrane would serve as the boundary, and everything in that boundary, within that boundary, you know, can be regarded as the organelles within that cell. So, for example, an animal cell, everything within the animal cell, are the organelles in that cell. So the same as a plant cell. So membrane just gives like a boundary. And then number two, organization and localization of function. What that means is that organelles help to organize functions such that the function of a particular, membranes help to organize functions such that the function of a particular organelle stays within that organelle, right? It doesn't, there is no crisscross, as it were. You don't have mitochondria performing the function of the nucleus because of the membrane. All of the enzymes, all of the markers of that organelle remain in that organelle because of the membrane that is surrounding that organelle. So, and then the function is specific to that organelle because of the presence of membranes. Membranes also help, with transport processes, and there are different types: active transport, passive transport, which we're not going to really delve into, extensively at this level. But you have, for example, if you look at this figure, you have potassium moving in and sodium moving out. So you have this sodium potassium pump. So membranes are where you have these pumps, these channels, that controls the entry and exit of some of these ions, and solutes, and then as well as nutrients. So nutrients move in and out of the cell, and membranes allow for this. You also have receptors that are present on membranes. Receptors are basically proteins, and they allow ligands, to bind to them. So here, these three, something that look like is like squares with a circle, like sharp edges, um, are receptors, and then the blue circles, the blue spheres, are ligands. So ligands bind, and then a chemical response um, is elicited. So we call that signal transduction, right? And ligands also, there are different types of ligands, which you would look into at higher levels. But for now, we expect you to know that on membranes, you have receptors that allow ligands bind to them. Then membranes also help respect to cell communication. So, when, for example, and that's the example I like to give, and when a male and a female comes together, they kiss themselves. There is exchange of saliva. You may not know, and that's why some people, you know, could even contract illnesses as a result of some of these things. So cells also exchange materials, they communicate to themselves um, when they come in close proximity with each other. So if you look at this part, you can see because of the presence of two membranes coming together, they are able to share materials, with each other. So some things can move from cell A to cell B, or from organelle A to organelle B. And that's what happens. So sometimes some proteins, for example, could be synthesized in a particular organelle and they are needed in another part of the cell. What happens is the membrane, or even in another organelle. So for example, mitochondria, you could have several mitochondria, in a particular cell, and then one maybe is defective and the other one is producing all it's supposed to produce. That mitochondrion is able to share, some materials with the neighboring mitochondrion, because of the presence of membranes. So it's the same as cells. So cells interact, they communicate through the presence of membranes. This slide just summarizes everything I have said. For emphasis, they define the boundaries of the cell and its organelles and act as permeability barriers. They serve as sites for specific biochemical functions, such as electron transport. So if you look at the mitochondria, you would see that there are different compartments, and I think that I have a slide on that, which I will show you in a couple of in a moment. These membranes have specific functions. So the inner membrane, inner mitochondrial membrane has its own function. The outer mitochondrial membrane has its own function. The matrix, you have so many things there. So membranes serve as sites for specific biochemical functions. Electron transport will not take place elsewhere other than the part of the membrane that is supposed to take place. So membranes also help in transport. I think I mentioned this. They help to transport proteins that regulate the movement of substances in and out of the cell and its various organelles. And then receptors, adhesion. Adhesion is when they are there. So membranes are the reasons some cells can adhere. We have what we call adhesion molecules, right, that help for these membranes to. Is the meeting not being recorded? Somebody is saying we should record. I thought I started recording. Yeah, meeting is being recorded. Okay. So, and then you have cell-to-cell contact and communication. All right, so let's still move on. In second year, you're going to be taught the models of the membrane, and there are different models, right, that have been postulated, but the widely accepted model is the fluid mosaic model, and that's what we want you to know at this level. This just basically means that the membrane, the structure of the membrane is both fluidic and a mosaic of proteins, right? So different scientists said it was only lipid. It was composed of only lipids. So there was something like lipid monolayer, so only one layer of lipids. But then some other researchers, continued to do more research, and they said, no, there isn't, beyond lipids, beyond the lipid nature of membranes, you also have proteins. And so they eventually said membranes comprises of proteins and lipids. But then later on, they also found carbohydrates, right? So eventually, when you talk about the structure of membrane, you would say that they comprise of lipids, proteins, and carbohydrates. And the most widely accepted model is the fluid mosaic model. And this is a figure showing what the fluid mosaic model proposed by Singer and Nicholson in 1972. This is what it looks like. So you have a phospholipid bilayer. So if you look at this figure, you can see, think of it like a bread sandwich. So, many of you who have eaten sandwiches would know what that looks like. So you have two layers. So the upper layer is the first layer, which is a monolayer, and then this other layer beneath. So that gives you a bilayer, two layers, two layers. So we refer to this as a phospholipid bilayer. And if you look at this figure, you would see blue spheres and some tails, some lines, right? I'll show you another figure. In fact, I just included it this morning so that you would have an idea. They are lipids, right? They are lipids. We call them phospholipids. And then these ones are proteins. So there are different types of proteins. You have integral proteins, you have peripheral proteins, you have lipid anchored proteins, not shown here, but I think I have another slide I will show you. The next slide actually shows that. But we need you to know that the membrane, so this line, and just so that you would have an idea, this black line here is actually this, what you can see here. So what you draw in the lab as a thick black line is not just a tiny line, it's a layer or a bilayer of lipids, proteins, and carbohydrates. So you have carbohydrates linked to these proteins as glycoproteins and carbohydrate linked to lipids as glycolipids. So just so you understand, do you understand what I've said? I've just explained about the membrane. If you understand, type yes in the chat box. No, yes, no, yes, no, please. Okay. So I said, we have more no's than yes. Are you sure you guys listened? Are you listening? Okay, so I said that this black line here, this membrane, so I'm just going to go over it again quickly. This black line that I said is the plasma membrane. And if you look at these other figures, so maybe I should go back. So this is the membrane here in a bacterial cell. This is the membrane here in an animal cell. Where is the membrane here? This is the membrane in a plant cell. This membrane that just looks like a line, surrounding the cell is not a line. And that's what I'm trying to tell you. So this figure even shows it better. This line here that we said has um, um, serves as a barrier that we said helps to with transport processes, help with signal detection. This is that line. That line is this. It's a sandwich, a bilayer of phospholipids, of proteins and carbohydrates. That's what I just said. So do you now understand? If you understand, those who said no now, only those who said no, I think they should type yes. So do you now understand what I was trying to say? Those who said no. Yes, good. All right. So don't think of the membrane as a line. Think of the membrane as this, a bilayer. And even that bilayer, we're going to look at it in depth. So this is that bilayer. And these guys, Singer and Nicholson, that's a typical exam question. Who proposed the fluid mosaic model, that explains the structure of the plasma membrane? A, Singer and Harrison, 1972. B, Singer and Nicholson, 1972. What's the answer, A or B? In the chat box, A or B, what's the answer? B. Okay, B, good. So, and then another question could be, which model explains the structure of the plasma membrane? Lipid mosaic model, A, B, fluid mosaic model, A or B? Let's see, A or B again. B, good. All right. So let's move on. Another question could be membrane comprises of A, lipids, B, lipids and proteins only, C, lipids, proteins, and carbohydrates. What would the answer be? A, B, or C? Let's see. A, B, or C? Good. C. All right. You guys are intelligent students. So let's move on. So this is that same plasma membrane, but this is another figure. And I'm going to show you three figures. So I'll show you the first one, but then this figure then shows you all of the type of membrane proteins. So I told you that you could have integral proteins, meaning that the proteins are embedded in the bilayer. So they are within. When you say something is integrated, like integrated science or integral, it is within, embedded within. When you say something is peripheral, you say periphery, or some people even say periphery, it's just at the side, loosely attached by the side. And then you can also have lipid anchored proteins. That is, proteins are lipids are the ones anchoring, carrying the protein. So this is what this figure shows here. So if you look at this, you would see the lipid bilayer. Remember, I told you it's a bilayer, like a sandwich. So you have the upper bread layer, and then the lower bread layer. And then this is the hydrophobic tails. So I'm going to be mentioning some things now that might be new to you, but just follow through. So you have the hydrophilic head and hydrophobic tails. and then you have this protein. So there are different types of integral membrane proteins. You have three broad classes. Listen, you have three broad classes of membrane proteins. Number one, integral membrane proteins. Number two, peripheral membrane proteins. Number three, lipid anchored membrane proteins. That's another typical exam question. You then have four types of integral membrane proteins. Number one, integral monotopic protein, single pass protein, multi-pass protein, and multi-subunit protein. Peripheral membrane protein, just one type. They are loosely attached to the side, of the membrane by electrostatic and hydrogen bonds. So these peripheral membrane proteins are usually attached to the membrane by electrostatic and hydrogen bonds. Integral membrane proteins, like I said, they are within the lipid bilayer. They are embedded within the lipid bilayer. And you have them sometimes spanning these are alpha helices. So at this level, I don't know if you have been introduced to the structure of proteins, but you have the primary structure, secondary structure, tertiary and quaternary structure. But we have alpha helices, beta pleated sheets, and all of that. But maybe I shouldn't bore you too much with that. But you have, single pass proteins, multi-pass proteins, meaning that they are spanning the lipid bilayer more than once. When you hear multi-pass means multiple pass, and then multi-subunit means that they have more than one subunit. I could give examples of each of these proteins. For example, single pass protein, an example I think is glycophorin, right? I'm not sure, but I think an example of single pass protein is glycophorin. An example of multi-pass protein would be what? Now, yeah, bacteriorhodopsin, I think, because I think that spans like 7 times. Yes, that's a typical example, bacteriorhodopsin. And then an example of a multi-subunit protein would be spectrin or even hemoglobin. It has multiple subunits. Right. So you also have peripheral membrane proteins. And an example of that one would be like ankyrin. But you then have lipid anchored proteins. Lipid anchored proteins. Let me just make a note of something because I need to double check that. You have lipid anchored proteins, and then we have two types of lipid anchored. And from the name, it means lipids are the ones anchoring, carrying them. So you have fatty acid anchor or GPI anchor. GPI is an acronym for glycosyl phosphatidyl inositol. Maybe I should type that in the chat box. List the types of integral membrane protein. They are shown in this figure. Glycosyl phosphatidyl inositol. GPI. To meeting group chat. Glycosyl phosphatidyl inositol. So that's the meaning of GPI. So it's also a type of fatty acid that anchors protein. It carries protein. Anyway, we also have beta barrel proteins, but not shown in this figure, but in year 2, we'll probably show you during that lecture. So this figure is from, remember that in the last lecture, I told you I got some figures from Dr. Adebeshi's previous slides. And I like this figure because it then shows you the labels. And I've deliberately put it, almost at the end of this plasma membrane part, because I wanted to have shown you like all of the other ones individually, and then you will now see all of these ones, holistically. So you have the carbohydrate chains. Remember, I said that the fluid mosaic model. So this is the typical fluid mosaic model. You have glycoproteins, you have glycolipids, you have the external membrane surface, the internal membrane surface. This is the inner surface, the side facing, the cytoplasm. So you have the cytoskeleton. You have lipids such as sterols, cholesterol. So in your body, many people will say, oh, your cholesterol level is high, your cholesterol level is low. If your cholesterol level is low, it will affect the membrane structure, because I told you that membranes are made up of lipids. And one important lipid in mammalian membranes is sterol, cholesterol. Particularly. Of course, it shouldn't be too much, neither should it be too less. So you can see cholesterol here labeled. You also have integral protein. So this is a peripheral protein. This is an integral protein. Where is it? This is a channel, membrane channel protein. So I told you that you could have channels that allow for the movement of substances, ion solutes into and out of the cell. So it could also be an integral membrane protein that allows. So you have the phospholipid bilayer. You can see it here. And then these are polar regions. Polar means they love water. So these circles, these balls, these blue spheres with the tails, or these ones here, these yellow ones with the tails, are actually this. So this is it. So if you look at this part of the slide, this one, you would see all of those things that made up the sandwich that we showed earlier on. All of the things that made up the sandwich that we showed earlier on. So you have a hydrophilic head and an hydrophobic tail. That hydrophilic head, so again, I've told you three things this morning. The line is not a line. It's actually a bilayer of all of this with a layer of, that's why it's a mosaic of proteins. So when you draw a mosaic in fine arts, those of you who did fine arts in high school, you know what a mosaic is. So that's why they describe it as a mosaic of proteins and fluid as well. So it has lipids, proteins, and then even carbohydrates. But when you look at even that lipids or phospholipid bilayer, you then see what looks like this. And even this, the hydrophilic head, is made up of choline linked to phosphate and glycerol backbone. And that's what makes up the polar head. The non-polar part, which is the tail, is an hydrocarbon tail. And the tail can be saturated or unsaturated. So from organic chemistry or chemistry in high school, you know that when you have more than one double bond or even one double bond, there is that leads to unsaturation of the carbon chain. And so that gives a double bond. So for example, you have alkanes, and alkynes. So when you have double bond, you call them alkanes, triple bond, I think alkyne. So in fatty acids, when the tails have one or more double bonds, it leads to what we call a kink. A kink is like a K, like K leg. So instead of it being straight, for example, if you look at this part, the left, you can see that there is no double bond here and it's just straight. But once you have double bond, at that point where there is a double bond, it branches out and then it forms what we call a kink. So the more double bonds you have, the more kinks you have. So please note the word. It's called a kink. Right. And so each phospholipids have a polar head and two hydrophobic tails. These tails are fatty acid chains, right? And like I said, they could be saturated or unsaturated. So let's quickly look at the endomembrane system because of our time. So that membrane that I showed you, Let me quickly check the chat box. That membrane that I showed you, please don't write your name in the chat box so that I'm not distracted by attendance. I'll pull it from Zoom. So the membrane I showed you also has, or is also linked to what we call the endomembrane system. And it includes the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, vesicles, vacuoles, and plasma membrane. 7. 1, 2, 3, 4, 5, 6, 7. 7 of them. And that's what we call the endomembrane system. So the endomembrane system is not an organelle. So you need to note this. It's not an organelle. So we're looking at cellular components, cellular structures, right? So some of the things we mentioned, for example, this endomembrane system, even though we are talking about it, is actually not an organelle. But then it is part of this of the cell. Now, this system carries out a variety of tasks, including the synthesis of proteins. So they help to synthesize proteins. This system, the endomembrane system, also helps to transport proteins into membranes and organelles or out of the cell. They help with the metabolism and movement of lipids, and then they help to remove poisons. So because of the lysosome, which is one of the functions of lysosome. So because the lysosome, which is an organelle, is part of this endomembrane system, this system works together. So think about it. Think of it like a post office, for example. In a post office, you go, you submit your parcel, somebody picks it up, packages it, puts it in an envelope, labels it, somebody else scans it, prints a QR code, places it on it. Somebody else, thinks that, oh, this paper is torn or the envelope is torn. They remove the torn envelope, which quote unquote can serve as poison, and then replace it, or they put it in another bag. They say, this bag is weak. They put it into a new bag because they have removed the former bag. So all of these things, eventually, until the parcel gets to where it is going, many people are working together. So the Golgi, sometimes we even refer to it as a post office. You have the AR where these proteins are synthesized. You have the rough endoplasmic reticulum and the smooth. So this figure just shows you what the endomembrane system looks like. So you have the nuclear envelope, and even the nuclear envelope is actually made up of membranes. So you have the outer membrane and the inner membrane making up the nuclear envelope. And that membrane is continuous, right, to the rough endoplasmic reticulum. The rough endoplasmic reticulum is the part of the endoplasmic reticulum that has ribosomes studded on it. So ribosomes, all these point dots, they are ribosomes that are present on the rough on the endoplasmic reticulum, and that makes it rough. The other part of the endoplasmic reticulum that does not have ribosomes is referred to as the smooth endoplasmic reticulum, and their functions are different. So the rough endoplasmic reticulum helps with protein synthesis. The smooth is the site of lipid and steroid, and sometimes some hormone synthesis. So you also have the part that we refer to as the cis Golgi. So remember I said that the Golgi is also part of the endomembrane system. So you have the Golgi, and so this is the nuclear envelope. This is the ER. This is the Golgi. So the side facing the endoplasmic reticulum is referred to as the cis Golgi, CIS. The side behind the endoplasmic reticulum, farther or farthest from the endoplasmic reticulum, is referred to as the trans Golgi. Please note, these are typical exam questions. So you have the cis Golgi and then the trans Golgi. So the cis Golgi is the side very close to the endoplasmic reticulum that is going to receive the oncoming vesicles coming from the endoplasmic reticulum. That's the cis Golgi. The side that would then bud off and send the processed and packaged vesicle to the plasma membrane is referred to as the trans Golgi. So if you look at this figure, you have the nucleus, you have the nuclear envelope, you have the the endoplasmic reticulum, you have the smooth ER. But please note, the smooth ER is not part of the endoplasmic of the endomembrane system. It is not part of the oh no, sorry, the domains. I've mixed it up. So you have the endoplasmic reticulum, and then you have the cis Golgi, and then the trans Golgi. So from the trans Golgi, you have the plasma membrane. You also have lysosomes, which is shown here. And then you have transport vesicles, various vesicles. So that's what I have emphasized in this bullet point. So nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, lysosomes, vesicles, vacuoles. You also have vacuoles. I don't know if this figure shows any vacuole. Is there any vacuole here? No, but you can also have vacuoles as part of the endomembrane system. But lysosomes collect some proteins that are not properly synthesized, properly folded, or that are not properly sorted and packaged, and then it destroys them, it digests them. So that's the function of the lysosome. But then when they're properly sorted in the Golgi, by the Golgi, they then get transported to the membranes where they are needed. Okay, quickly, because of our time, we have some membranes that have some organelles that have two membranes. For example, the nucleus, mitochondria, or plural mitochondria, the chloroplast, they have double membranes. Some organelles have single membrane, one membrane. So an example is lysosome, peroxisome, vacuoles, endoplasmic reticulum, and the Golgi. Some organelles do not have membrane, and I think I mentioned this, for example, ribosome. Some people also say cytoskeleton, is an organelle or maybe a cellular structure, and they also do not have membranes. In fact, later on in year 2, you will look at the what the cytoskeleton is made up of: rigid fibers, proteins. For example, microtubules, you have alpha tubulin, beta tubulin, all of those kind of things. So, but they lack membranes, right? They do not have membranes. So these are likely questions. You could just have a list of all of these and then be told to match correctly. So which of these is mismatched? So you should know. Ribosomes do not have membranes, but it is an organelle because it has its specific function. Okay, so what is the ER? Let's look at some of these organelles individually. So we've looked at the plasma membrane. The next organelle I want to look at is the endoplasmic reticulum. And endoplasmic simply means within the cytoplasm. Within the cytoplasm. Reticulum is a Latin word meaning network. So the ER, also known as the endoplasmic reticulum, is a continuous network of flattened sacs, tubules, and associated vesicles that stretches throughout the cytoplasm of the eukaryotic cell. It is continuous with the outer membrane of the nuclear envelope. I told you that the nuclear envelope has two membranes, the outer membrane and the inner membrane. So the ER continues with the outer membrane of the nucleus. That's why it's also part of the endomembrane system. Now, the membrane-bounded sacs of the ER are called ER cisternae, plural. The singular is cisterna, cisternae. And then the space in each of these sacs is called the lumen of the endoplasmic reticulum. I've mentioned this earlier on, that the rough ER, because of the presence of ribosomes, is the site for transport of helps with transporting proteins that are made by the ribosomes. So ribosomes actually help with protein synthesis, but then this happens, and these proteins that are synthesized by the ribosomes are then transported by the RER, while the SER, which lacks ribosomes, is a site of lipid and steroid synthesis. So this is the ER. So if you look at these spaces, these lines are the cisternae. You can see it labeled. One of them is cisterna. So sometimes you can have 3 to 8 cisternae, depending on the need of the cell. And then you have these ribosomes studied on them. So ribosomes on the ER, protein synthesis and transport, basically. The smooth ER, lacking ribosomes, lipid synthesis, steroid synthesis. You then have transitional ER, and that's what I mentioned earlier. So it's a domain of the endoplasmic reticulum, and this is the nuclear envelope, which is continuous with the endoplasmic reticulum. The outer membrane of the nuclear envelope is continuous with the endoplasmic reticulum. So when we talk about cisterna, so this is cisterna. Actually, this label might be wrong because it is speaking to, and we've said that cisternae is plural. Cisterna is one. So, but if you look at this cisternae, it is one way to look at the endoplasmic reticulum. But the endoplasmic reticulum could also be seen morphologically as vesicles and as tubules. So I'll show you. So these are tubules, the endoplasmic reticulum, which is smooth ER. These are cisternae, right? You can see these are cisternae, which is still the endoplasmic reticulum. And then you have vesicles coming, pinching from the ER, and it is still the endoplasmic reticulum. So if you see a figure that shows transport vesicle, this vesicle itself is an ER, is an endoplasmic reticulum. This tubule, like tubes, is also an endoplasmic reticulum, and then this cisternae is endoplasmic reticulum. So these are the ways or things that we see under the microscope, you know, or I mean, when you're describing the morphology of the endoplasmic reticulum. So 3 of them will be correct. So it's not just cisternae, not just vesicles, not just tubules. Sir, please, what is, I could see one, sir, please. The names have covered your question. What is paroxysm? We would get to paroxysm in a moment as well. So let's move on quickly because we still have a lot to do and I only have one more lecture. So I said that there are 5 subdomains of the endoplasmic reticulum. The first one is the nuclear envelope. The nuclear envelope. The second one is the rough endoplasmic reticulum, then the transitional ER, mitochondrion-associated membrane, and the endoplasmic reticulum Golgi intermediate compartment. 5 of them. You're expected to know this. And the reason I've brought this is because some of you will not be coming back to take other biology courses. So it's important that you note. So if you look at this figure that I showed earlier on, you have the nuclear. So I said there are 5: nuclear envelope, rough ER, transitional. So this is the nuclear envelope. It's a domain. You have the rough ER, another domain. Note, the smooth ER is not a domain. Instead, the transitional ER is a domain. The transitional ER is a domain. And then you have the mitochondrial associated membrane, which is not shown in this figure, but there are other figures that we will show you in 3rd year that you will see the mitochondrial associated membrane. Then you have what we call the ERGIC, EGIC, or endoplasmic reticulum Golgi intermediate compartment, which is an intermediate compartment between the endoplasmic reticulum and the Golgi. Remember when I told you that the side facing the endoplasmic reticulum is called the cis Golgi, and the side behind, which will be somewhere here, is the trans Golgi. So between the endoplasmic reticulum and the cis Golgi, you have sometimes an intermediate compartment, not complex, intermediate compartment that collects vesicles coming from the ER and serves as an intermediary between the ER and the Golgi. Does 1 or 2 things before sending it to the cis Golgi. And then sometimes from the cis Golgi as well, you can have some vesicles going to the endoplasmic reticulum, but sometimes it also goes through the intermediate compartment. So the Golgi complex was named by the scientist Camillo Golgi. He was an Italian biologist, and he described the Golgi, also known as the Golgi apparatus, in the year 1898. And it's a series of flattened membrane-bounded sacs. So Golgi also has cisternae. So note, is similar to the ER. It has cisternae, continuously being formed at one end of the stack and budded off as vesicles at the other end. The same way you have vesicles budding off from the ER, Golgi complex also has vesicles that can bud off. And so you could have 3 to 8 cisternae per stack. And like I said earlier on, the number of every organelle usually depends on the type of the cell and the need, the metabolic activity of the cell, depending on what so if the cell needs what will I use now as an example? ATP, for example, there will be more mitochondria. So, for example, in your muscle cells, you have more mitochondria than maybe elsewhere. The sperm cell in a male also requires more mitochondria, and I'll show you in the next few slides. So, depending on the need of ATP, you would have more mitochondria there. And remember that I showed you one table, that showed that in the liver cell, for example, you have, several mitochondria there as well. So it depends on, because there's a lot of metabolic activity going on and you need cellular energy, right, which is produced by, so there will be more. So it's the same thing. Depending on the metabolic activity of the cell, the number of the Golgi stacks might increase or reduce. So in the Golgi, 2 things happen. Proteins from the ER. Remember we said that the ER helps with protein and lipid synthesis, the rough ER proteins, the smooth ER lipids. But these proteins themselves are produced in the ribosomes, which is the machinery for the synthesis, but they are present on the endoplasmic reticulum. And that's why the ER is associated with protein synthesis. But then they also help with lipid synthesis, and the Golgi then sorts these proteins. Okay, which protein is going to where? So that's why I told you it's like a post office. Which protein is going to Iyanokpaja? Which protein is going to Yaba? Which protein is going to Lekki? So the Golgi is the one that sorts all those proteins. It packages them. It says, okay, come into this vesicle. Those of you going to Lekki, enter this vesicle. All of them will enter the vesicle, and then it will pinch off and start moving. Those of you going to Yaba, enter this, is like a bus. So it carries it. So that's what the Golgi does. It packages it for transport to their proper destinations inside the cell or outside the cell. Remember that you need proteins, enzymes. Most of the things going on in your body would require one enzyme or the other. Now imagine this protein is synthesized in the ER. These lipids, as even when I told you your membrane comprises of lipids, this lipid in itself is synthesized by the smooth endoplasmic reticulum. Now, this smooth endoplasmic reticulum will be somewhere far, but then the membrane, this plasma membrane, for example, is somewhere else. So the Golgi is the one that would help to collect the lipid that has been synthesized by the smooth ER and then send it to the plasma membrane through the trans-Golgi network that I showed you earlier. So Golgi complex also helps to make lysosomes, and it plays an important role in membrane and protein trafficking in eukaryotic cells like the ER. So Golgi also helps, please note. Remember I told you that later on we might look at how organelles, and that's one of the things we're interested in cell biology. That's one of the things we're interested in cell biology, how organelles are inherited, right? How organelles are produced, organelle biogenesis. So Golgi is one of the organelles that helps to produce lysosomes as well. So this figure just shows the figure of of a Golgi complex. So like I told you earlier on, you have cisternae, which are plural, more than one cisterna. You have transition vesicles coming from the ER. This is the cis-Golgi network. Then TGN, the trans-Golgi network. These vesicles will pinch off like you pinch somebody. It will pinch off from this part after it has, so it will receive the protein that has orishirishi that needs to be removed. So think of it like you want to go for a haircut, right? So the cis Golgi network is the part that receives the rough looking guy, and then it packages it, removes everything that needs to be removed, and then it is clean, looking clean and sharp. And then the trans Golgi network then pinches off another vesicle and sends it to this membrane, the plasma membrane, or to the membrane of any other organelle, maybe a mitochondrial membrane in the cell. I believe you understand because I have explained it over and over again. I have tried to repeat severally intentionally so that you will get the gist. But just to be sure, if you understood, type yes in the chat box so that I'm also sure that you got the gist. Yes, good, good, good. All right, so we just move on. Those of you that said no, you watch the recording because we don't have time. All right, yes. So we move on to the next organelle, which is the mitochondria, because we have only one more lecture. So I have to finish my slides so that the next lecturer will come in. And I think you have your test coming up very soon. So what I'm going to do is I'm going to send my slides after today's lecture to your groups so that you would read up onto the part that I stop. So your questions will not go beyond this part. I think I still have like 10 minutes. So I hope to finish this slide today. But please try to read up until maybe wherever we stop today. And I think that maybe we should stop after mitochondria, or we can also look at the nucleus. Okay, so, but you won't be asked questions of what you have not been taught. That's the gist that I wanted to pass across during your test. If I get to set all my questions for your test, before I finish my lecture, but I think that I might have finished. I think your test is next week. Oh, so I still have one more class. Okay, no, I'll be fine. So maybe I shouldn't send it today. I'll send it at the end of my next lecture on Friday. All right, so the mitochondrion, plural mitochondria, right? It has, the term is from a Greek word or from two Greek words, mitos and chondrion. Mitos means thread, chondrion means granule, or grain-like grains, like grains or granules. And from secondary school, we refer to the mitochondria as the powerhouse of the cell, the powerhouse of the cell. And the reason it is so is because it generates ATP. It's the primary site of ATP synthesis. And ATP is synthesized from the oxidation of carbohydrate, fats, and amino acids. So when you eat, remember there is glycolysis. Glycolysis produces ATP. There is the Krebs cycle. You also have some form of production of energy. But then you then have the electron transport chain, which occurs in the inner mitochondrial membrane, right? Which is where and maybe, I don't know if I have the slide here. Okay, I'll probably put it before Friday because I need to show you. Again, you see that membrane structure and all the complexes, the proteins in the inner mitochondrial membrane that helps with ATP production in the, via electron transport. So, but just note that the mitochondrion is the site of ATP synthesis. And then they have two membranes that differ in there. Remember when I gave you examples of organelles that have double membranes, mitochondria and chloroplast were listed, as well as the nucleus. So the inner membrane is also present, and it contains important enzymes of energy metabolism. The matrix also contains enzymes, and I'll show you what the matrix is when I show you the figure of the structure of the mitochondria. Please note, when I told you earlier on that all cells die, I said that they can either die through apoptosis or necrosis. At this level, note that the mitochondria, there's a mitochondrial pathway of apoptosis. Those of you that would do year 2 cell biology or cell physiology will teach you apoptosis, will show you the intrinsic and the extrinsic pathway. The intrinsic pathway is the pathway of apoptosis via the mitochondria. So you would see release of cytochrome C from the outer mitochondrial membrane, blah, 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 blah. At this level, don't worry too much about it, but just note that this cell death that I told you happens automatically, programmed cell death. When a cell grows old and is destined to die, can the mitochondria has also been implicated. It helps to remove unwanted cells. Sometimes you need apoptosis to happen. For example, when a woman is pregnant, if you look at the figures, so those of you in medicine, you look at the figures of a pregnant woman with a fetus, you see that sometimes the figure looks like a lizard, like a tadpole initially, and then later on, it continues to look until it now looks like a baby, right? After maybe about 30 something weeks. What has happened is that apoptosis continues to happen, continues to remove unwanted cells, from the limbs, from the legs, from the eyes, and then the baby is fully formed. So apoptosis is something that is supposed to happen, or else there'll be a problem. So sometimes what happens in cancer is that there is no cell death. So cells are dividing uncontrollably. Whereas some of those cells are supposed to die, but then you have uncontrolled cell division. They are not dying. Apoptosis has been altered because there is a mutation in the gene that controls cell cycle progression. Anyway, all of this is advanced biology, but please, I've just said all of this so that you have a mental picture of this. Mitochondria reproduce by binary fission. This is the size of mitochondria, similar to the size of bacteria, 0.5 to 2 micrometer. 0.5 to 2 micrometer in diameter, and the length is about 7 micrometer in length. So this is diameter, 0.5 to 2, and then this is the length. Mitochondria looks like a sausage. So those of you that like hot dog, sausages, that's what mitochondria. But there are other shapes. So it could look like a rod, which is more like the sausage shape, like a vesicle. So you have some mitochondria that are vesicular in shape. Some are filamentous, like thread-like. Remember that even the word granular, grains, they look like grains. So if you think of rice grains or any other grain, food grains. So mitochondria also looks like that. Sometimes they look like a racket, sometimes they are round or ring-shaped, and sometimes they look like a club shape. So this figure on the right shows a rat's primary spermatocyte. And then the mitochondria of the rat is granular. For example, if you look at this figure, it's granular. For turtle liver cells, it could be round or club-shaped. And then in mammalian kidney cells, you can see it looks like a thread-like, like filament, filamentous. So it's just like a filament, elongated. So in mammalian kidney cells. So in various cells, you have different shapes of mitochondria. And all of these shapes have just been summarized here. I told you earlier on that depending on the need, the metabolic activity of the cell and ATP needs, the number of mitochondria will increase. So for example, in your muscle cell, you need so much mitochondria because you need ATP, right? And that's why people who have muscle problems, weak bones, all of this osteoporosis, some of them have problems with their mitochondria, their mitochondrial diseases. So they are not producing ATP, right? And that's why they have to, do maybe physiological exercises and all of those kind of things to kind of increase and help and take some drugs that can help, with the production of ATP via the mitochondria. So, but your muscle cells have mitochondria and the sperm cells. So some sperm cells as well. The reason some sperms cannot swim is because of the problems they have with their mitochondria. So if you look at the structure of this sperm cell, you have the nucleus, but then in the tail region, you have the mitochondrion. And this mitochondrion, you also have 9 2 central doublets and 9 singlets. Yeah, that's what it's called. Yeah. Okay. Yes, it's even here 9 plus 2. So, but anyway, I don't know if I want to bore you too much with all of this gist, but you have the mitochondrion in the mid piece of the sperm tail that helps the sperm, the flagellum, Remember, we talked about cell motility. So this is the sperm cell. This sperm has to swim to the egg cell to fertilize the egg cell. And then, how would the test be? Dr. Fakarede will communicate to you. This sperm cell helps, this mitochondria helps the sperm cell to swim to where the egg cell is. And of course, this first sperm cell that gets to the egg cell fertilizes the egg, and the zygote is formed, blah, 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 until you have a baby. But sometimes you have men with fertility issues. Their sperm cells are not swimming. So those of you who do your IT in the hospital, you see that the sperm cells are not, some of them do not even have tails. And when they do not have tails, so you just have sperm head without tails, because there are no tails there, the sperm head cannot swim to where the egg cell is. You have a lot of problems. Some sperm cells, you don't even have head. It only has tails, and the tails is just there, the head is somewhere else. And so you're wondering how would but the mitochondria helps because this sperm cell needs ATP to be able to swim, via flagellar movement to where the egg cell is. So this is, so every sperm cell requires more mitochondria in the mid piece of the sperm cell. The same way muscle cells also require. So this is the structure of the mitochondria. We don't have time. Wow. I thought that I will finish this slide today. So the mitochondria, you have 4 major parts: the outer membrane, the intermembrane space, the inner membrane, and the matrix. OMM, outer mitochondrial membrane, intermembrane space, IMM, inner mitochondrial membrane, and then the matrix of the mitochondria. So if you look at this figure, the space here is the matrix. The first membrane, the outer part, maybe this figure shows it better than the electron micrograph. So this is an electron micrograph, right? So, okay, here they just even lumped it together, inner and outer. But if you look at this, and then you have the cristae. So if you look at this one, you see the outer membrane, then you see the inner membrane here, and then the space between the inner membrane and the outer membrane is the intermembrane space. Remember, I told you that these membranes are not just lines like this. Like this one now is showing orange line, but it's not just orange line. In fact, if you look at this, you can see 2 lines, 2 black lines, and that's to tell you it's a phospholipid bilayer, which is what I showed you earlier on when I started with membrane. So, and then if you look at the inner membrane as well, you can also see 2 lines there. So it's also a phospholipid bilayer, talking about the fluid mosaic model of the membrane. Do you have another class after now? Yes or no? One person should just type. Once you see the first response, it's okay. Yes, you have another class. Okay. I wanted to continue. But anyway, it's 9 o'clock already. So maybe we'll just stop here and then we would continue so that you join your next class. If we have one question, maybe we can take one question before 9. Do we have one question? No. Okay. I don't see any questions in the chat box. Can somebody help me also check the YouTube page if we have any question there? No. All right, so we'll just continue from the mitochondria next quick. Please, can you send the slides today? Okay, maybe I should, because I only have 4 more slides to go in this part. So the Friday slide will be different from this one. So that will be slide 2. What is ATP? You should know what ATP is now. ABBA. Yeah, in year 1, adenosine triphosphate. Somebody already mentioned it in the chat box. All right, so I would send the slides to your class rep today so that those of you who want to start reading can already start. Maybe before the end of today, I'll send it. So I'll just update it with maybe 1 or 2 more things, and then so that I'll know that I'm done with this part. And then by the next lecture, we'll look at chloroplasts and maybe briefly photosynthesis and take more questions. So there is also a table that I have that summarizes all the functions of some of these cellular components and organelles, which would also help. But that will be all for today. Thank you and see you. How does the body tell the protein where to go? There's a lot. So there's a lot. That's why I told you, you have ligands, you have receptors. So receptors are specific, right? So the protein knows the receptor. So there is a receptor. So for example, there is trail. Trail is the protein or the ligand. Then trailer with an R is the receptor. So trail goes to trailer. TRAILR. So all of these things, so the ligand is there, the receptor is there, so they're specific. But there are so many things. So they're what called peptide determinants. These things are things that you will learn, in higher levels. But for now, don't worry your head too much about it. If you're interested, come to cell biology or biochemistry. I think they would also teach you there. All right. Have a good day. I will just stop for now.